Good evening, ladies and gentlemen. My name is Sarah Sanchez, and I want to welcome you to a very special 1869 program directly with a group of 3 scientists who discovered DNA and its structure. We have here in the studio Dr. Miguel Pulido, who discovered nitrogenous bases. Then we have Dr. Daniel Barriga, who discovered the phosphate and sugars of DNA. And then we have Dr. Jaime Vence, who discovered the DNA structure and the different types. DNA is self-replicating genetic material that can be found in all living organisms. In simpler terms, it's a carrier of genetic information. Apart from being responsible for this genetic information, it also plays an important role in the production of proteins. Dr. Miguel Pulido is here with us today in order to present his discovery of nitrogenous bases. We have discovered that there are 4 nitrogen-containing nucleobases that can be found within DNA. First we have adenine, guanine, cytosine, and thymine. Adenine and guanine are known as purine bases, while thymine and cytosine are known as pyrimidine bases. Nitrogenous bases form base pairs between guanine, cytosine, adenine, and thymine. These then form hydrogen bonds between the opposing strands of DNA and form the appearance of a twisted ladder of the double helix in DNA. Dr. Daniel Barriga here has discovered sugars found in DNA. The sugars found in the nucleic acids of DNA are known as pentose sugars. A pentose sugar has 5 carbon atoms. The combination of the base and the sugar is known as a nucleoside. The sugar found in DNA is known as deoxyribose, which is a modified sugar that lacks 1 oxygen atom, hence the name deoxy. The difference of the absence of the oxygen atom is very important for the enzymes that recognize DNA. These 2 important discoveries have paved the way for discoveries of different structures within the different types of DNA. This discovery was led by Dr. Jaime Vence. Dr. Jaime Vence has discovered that there are 3 different types of DNA. First, we have aDNA, which is a right-handed double helix strand with very short distances between its base pairs. Then we have bDNA. This one is the most common form present in most types of DNA. This has the very classic appearance of a right-handed double helix structure. Lastly, we have zDNA, which has a very different structure. This has 2 strands that coil in a left hand and have a zigzag shape, hence its name. And with these new discoveries, we're ending our program, leaving off to see what our group of scientists discover next. We thank you very much for tuning in and hope you have a great night.